guess we'll get started for today. So if anyone hasn't given me your homework here, please make sure we have that. We'll be giving them to the grader right after class, so it's important we have all of them here now. I'll just briefly mention, we're having a couple of technical problems. This screen seems not to be working. I hope everyone can see the screen that I'm standing right in front of. And we're also having some problems with Blackboard right now. Basically, I can't upload anything for Blackboard, which is why today's lecture notes aren't there. So I sent them an email to try to get that fixed. But hopefully, you guys will all see this. So that will be fine for now. But basically, what we're going to be doing is finishing up our discussion of sequence searching and alignment and trying to get into some of the theory behind how the tools for this problem work, as well as getting into a bit of the hands-on work of actually trying some of these tools and seeing how we use our knowledge of how the algorithms and statistics work to use these effectively. So before we get to that, are there any questions about anything up to now? Any issues from this past week? Okay, so just to review where we left off. Last time we had covered the basics of what is alignment, what is an alignment score, and a scoring function. So we should have a basic idea of what it means to align two sequences, how you would score these alignments based on, let's say, matches, mismatches, and gap scores. We should have some idea of the differences between global, local, and semi-global alignments. And can anyone tell me which of those three is this one? Let's illustrate it at the top here. Yeah, this is a global alignment because we're penalizing for gaps or extra overhanging pieces in both sequences. What's this one? Yes, that's local. We're not penalizing for gaps in either sequence. We're looking for a subset of both sequences that match, penalizing for n gaps at least. And that would leave this as a semi-global alignment. We're penalizing for the overhanging end in one of the sequences, but not the other. We saw the concept of alignment statistics, so how an alignment score is used to assess the statistical significance of an alignment. You should all understand at least the basics of how this is done and know that it gets quite a bit more complicated in some cases when we start worrying about gaps and worrying about issues of, for example, multiple hypothesis correction in local alignments. But basically, this gives you the central idea, and you should all be aware of that by now. And you should be aware of what a substitution matrix is and how that becomes important when we're talking about especially amino acid alignments. So are there any questions about any of those issues at this point? Okay, the last thing we covered is multiple alignments, so you should know essentially what the difference is between a pairwise alignment of two sequences and a multiple alignment of 3 or 4 sequences. And that's where we get into the issue that we hadn't covered last time, which is how do we actually get these alignments? So I want to talk a bit about alignment algorithms. And I know in this class I don't assume you've had any computer science or any algorithms training, so we can only go so far with this, but I want to give you at least a high-level intuitive feel for how you would create a computer program that does this, because it's often important in understanding what you would do with these kinds of alignments and how you would interpret the results or choose an appropriate alignment program. So if we're trying to align two sequences, it's actually possible to find absolutely the best possible way of aligning two sequences up to fairly large sequence lengths. And that's done through a class of algorithm called a dynamic programming algorithm. So if we're not worrying about gaps, it's pretty easy to figure out how to align two sequences. You can just take the sequences, slide them against one another in all possible ways. There aren't that many possibilities. But when you start worrying about gaps, it becomes a hard problem because the number of ways you could insert gaps in your sequences will blow up exponentially in the lengths of the sequences. So it's not obvious that you can find the optimal way of inserting gaps in two sequences to align them, but that's where a bit of algorithmic theory becomes important in showing that this is in fact a solvable problem. At very high level here, what this is doing is using a kind of strategy for algorithms known as a divide and conquer strategy, which basically means that we take a problem that looks hard, that's maybe not obvious how to solve, and we try to find a way of expressing it in terms of slightly easier problems. And in this case, what this is showing is something called a recurrence relation, which is a way of asserting that it's possible to solve the problem of aligning two sequences by considering the problems of aligning slightly shorter subsets of those sequences. In particular, if we have sequences A and B, you can find the optimal way of aligning the first I bases or residues of A with the first J bases or residues of B in terms of best possible alignments of slightly shorter versions of those sequences. So the best way of aligning the first I bases of J and the first I bases of A and the first J bases of B is either align the first I minus 1 and J minus 1 optimally and match the next two bases, or align the first I minus 1 and J minus 1 optimally and throw in a mismatch if those next two bases don't match, or align the first I minus 1 of A to the first J of B, throw in a gap, so you're saying that the next base of A doesn't align to anything in B, or vice versa, throw in a gap in B. One of those four has to give you the optimal alignment, and once you observe that, you can come up with a way of optimally aligning sequences of quite large lengths by simply starting with the smallest pieces. Start by aligning one base at a time, then use that to figure out how to align two, use that to figure out how to align three, and basically build up an entire solution. That's basically the idea behind a dynamic programming algorithm, and that gives us a way of optimally aligning two sequences. So for those of you who have taken an algorithms or even a basic programming class, this recurrence relation should give you almost everything you would need to actually write your own sequence aligner. But for the rest of you, I think it's important to understand that this is a solvable problem and to understand some of these basic paradigms of how you solve a hard problem. But don't worry about it if you don't know offhand how to write a computer program to code this. Hopefully by the end of the term that'll be clear to everyone. So there are a couple of versions of these dynamic programming algorithms. It's known as the Needleman-Wunsch algorithm for global or semi-global alignment and the Smith-Waterman algorithm for local alignment. And these algorithms were what really made it possible to start working with sequence databases in a serious way. So for a long time, these were the ways you would do this, and these algorithms were usable if you were talking about sequences of maybe 1,000 or so nucleotides. If you had a query of 1,000 nucleotides and you needed to align to a database with the order of a few thousand nucleotides. Unfortunately, that's not good enough if we want to start talking about modern databases, which have a lot more than a few thousand nucleotides in them. Before I get to what we actually do for the most part today, are there any questions about any of this? Okay, so in a modern database, we generally would have a lot more sequence. Those dynamic programming algorithms guarantee you get the best possible way of aligning relative to a given scoring function, and the time they take is approximately proportional to the product of your query sequence and the size of your database. So that is manageable if you have a very small database. It's not manageable with a modern database that might have exabases of sequence or even petabases of sequence. That's too much to deal with. And so you tend to need faster algorithms. So what's often done in practice is actually not to use these exact algorithms, the ones that guarantee you get the best answer, but to use heuristic algorithms that will usually give you the best answer. They might miss an answer that is the best one. They might get a suboptimal one, but they will do it much faster than those dynamic programming algorithms. And the most popular of these algorithms is something known as the Basic Local Alignment Search Tool, or BLAST. So BLAST is the main way people search for sequences in these databases. It's a more complex and in some ways less elegant algorithm than the dynamic programming ones I told you about. It's possible it will miss the best alignments in some cases, but it is many times faster when you're working with a large database. Now, there are some details we need to know about how BLAST works if you want to use it effectively, so I want to briefly describe how the BLAST algorithm works. So the basic idea behind BLAST is to use small sequences known as k-mers or words, and a k-mer just means a string of k nucleotides or k amino acids. So a 4-mer would be 4 nucleotides in a row, a 10-mer would be 10 in a row, and so forth. And what BLAST does is construct an index of all the k-mers in your database. So it runs through your database and tries to find from all your sequences all the strings of whatever length of k you're looking at. And it uses those to search for short alignments. So this is another version of that divide and conquer idea, that we can take the hard problem of building large sequence alignments, and in this case, try to turn it into the easy problem of building short sequence alignments. Because it's really easy to find short alignments in a sequence, and it's really easy to find exact or ungapped alignments. So if we can take the gaps out and we can look at only short sequences, then we can do this very quickly. And BLAST uses that to kind of get the search started by looking for these short alignments that are known as seeds, and then do a slower alignment around each of these seeds to see if we can extend that into a really high quality alignment. So that's the basic idea. And to illustrate how this works, I'll just take a couple of example sequences, and we can see what this might look like. So if we take our database sequence, what we would assume is that before you start searching against that database, you have run through and built your k-mer or word table. So we would take the database sequence and we would go through, and if we were using a k of 4, so we were looking for 4-mers, we would look at the first 4-mer in the sequence. And we would put an entry in our database that says this sequence, CCAG, is found at position 1 in our database. Here I'm assuming the database just has one sequence, but it's pretty easy to generalize many of them. And we could look at the next 4-mer, put that in our database, the next one, put that in our database, and so forth. Sometimes you would see that a k-mer would occur more than once, so HECT would occur at 2 different positions in this database. You just keep track of all of those. So is this clear to everyone? All right, so this would then be sitting around. You would pre-compute this before anyone has gone and started searching against your database. And then when someone came along with a query, what you could do is then look at the k-mers in the query. So we could notice that the first k-mer in the query shows up in our table. It's very easy to quickly look up an exact match in a table like this. And we could see that that occurs at 2 different positions in our database. So we could try saying that there is a potential alignment at those 2 positions in our database. Some of the following k-mers that we would get out of here would then show up one after another, starting with those same seed alignments, and you could use those to build on the seeds to effectively get the longer seed. And occasionally we might get alignments that occur in different positions. We might have that this is another example of this pattern and gives us a couple of other possible seed alignments that might be starts with good alignments or prior to our database. So that is the kind of fast pre-computation stage of BLAST. And the important point is that this is much faster than trying to optimally align the entire sequences. It's fast to build this table, and it's fast to look for these little k-mers and see where they are in a table like this. Any questions about that? And so if you're doing amino acid sequences, it's pretty much the same. You do have to consider some other issues that become important, very analogous to the issues we had to worry about in scoring amino acid alignments. And one important thing we need to pay more attention to is that it's often not enough just to record the k-mer in our database. So if we have a k-mer, let's say SRK, if we're using 3 mers in our amino acid sequence search, then we need to worry about the fact that you might not be able to find a good match to this protein unless you take account of the fact that there may be chemically similar k-mers found in analogous sequences that you also want to consider potential seed matches. So if we see an SRK, we might say that we want to recognize SRKs as a possible match to this in a query sequence, but also TRK, that might be similar enough, or SKK or SKR. So what we'll do when we see this in building our k-mer table is we don't just throw in SRK, we also try to create a neighborhood of similar k-mers, and we'll throw all of these into our table so that we'll recognize them when they come up in queries. Basically, essentially we want to throw in anything that is close enough that we think it would make a good match, or at least a good beginning of a match. Otherwise, it's pretty much the same. And actually, this comes up also in nucleotide searches. It's just less important than you worry about this in nucleotide searches. Any questions? Okay, so that basically is the protein blast idea. And once we've gone through this, we would end up with a set of seeds. And what we hope is that there aren't that many seeds. The important thing behind the blast search that lets it work is that if we have a big enough seed and the seed is specific enough, then by random chance you're not going to get a lot of seed alignment. Maybe you'll get a few by random chance that aren't actually part of a significant alignment, but you hope you don't get that many, because what you're then going to do is look at all of the seeds and try extending them. So try running your algorithms, a more exact one, so similar to the dynamic programming algorithms, just locally in the vicinity of that seed to try to extend it in one direction or the other, build together different seeds, throw in gaps if you need them, and get a really good alignment around that seed. So essentially, this is going to work as well as the dynamic programming algorithms, as long as your seeds are able to pick up the good alignments. And that won't always work. There can be cases where you have a good alignment in your sequence and you actually don't have a single good seed in that alignment. Can anyone think of how that might occur? How might you miss a good alignment with this approach? Well, one example might be, let's say we've got a really long alignment where every 10 nucleotides is a mismatch. So you have 9 good matches in a row, then a mismatch. 9 good, then a mismatch. If you have that over and over, that may be a very high quality alignment, so something that has very strong statistical significance, but if your k-mer length is 10 or more, you're not going to find it. You're going to get hit by that mismatch on every possible k-mer, so the BLAST algorithm could miss that really good alignment. So that's kind of the trade-off with these BLAST approaches. You may miss something that's good, but it's unlikely you will unless you have a very large word length. Any questions about this? Now, I will just point out that even these BLAST approaches are too slow now for a lot of the kinds of sequence we look at. So when people are looking at aligning to databases of sequence coming off the modern next-generation sequencing machines, even this kind of thing is going to be too slow when we're worrying about maybe aligning to genomes or aligning to large sets of data, like a large RNA sequencing data set against a genome. There's simply too much data, and it's now an active research area. There are faster algorithms that also can potentially miss some good alignments, but this is something people are working on today to try to figure out how to speed up alignment enough to work with the sizes of data sets we have today. But still, most people use BLAST for these kinds of things, and it usually does what you need for most of these applications. And any questions about that? All right, so there are a number of variants of these BLAST algorithms, and it's worth being aware what these are. So when we get into the BLAST tools, you'll see that there's one that's called BLAST-N, that's nucleotide BLAST. And what that does is take a nucleotide query and search it against a nucleotide database. So there will be a database of nucleotides, let's say genomic sequences. They'll build the k-mer table from the nucleotides, and then you take your nucleotide query. It will search for matches in that k-mer table in your query and do the BLAST algorithm like I described for nucleotide BLAST. There's a BLAST-P, which is protein BLAST, which is the analogous thing with the amino acid alignments. And there are also translated versions of BLAST. So you may be interested in looking at a nucleotide query matched to a protein database. So maybe you've done an RNA sequencing experiment, you've got lots of RNA transcripts, you want to figure out which ones code for proteins or what proteins. You might want to go to a protein sequence database, search your RNA query against it, and then to do that, what this is going to do is translate the query. So it's going to take your query sequence, which is nucleotides, and it will try to convert it into an amino acid string in all 6 possible reading frames. So one reading frame would be to say that we will assume nucleotide 9 and so forth. That's one reading frame. You can also assume 2, 3, and 4 are a codon, 5, 6, and 7, and so forth. And you can assume 3, 4, and 5, 6, 7, and 8, and so forth are codons. So that gives you 3 ways of translating it into amino acids, and then you can search all of those against your amino acid database. I said there were 6 reading frames, though. What are the other 3? Yeah, so you would also do the same thing in the reverse complement strand of your query. So that would give you matches of nucleotides to proteins, and you can also do the reverse. Take a protein query against a nucleotide database, and in that case, they would pre-compute the translations for the nucleotide sequence in all 6 possible reading frames, and have the Kamer table set up already for those 6 translations. There's one more common variant of BLAST that's called tBLASTX. And tBLASTX takes a nucleotide query against a nucleotide database, but translates both of them. So it translates the nucleotides and it translates the queries in all 6 possible ways, and then does basically all 36 possible ways of aligning those against one another. Can anyone think of why you might want this? Why not just use BLASTN? You're creating a lot more comparisons here, and more comparisons is generally a bad thing with string searching because you need more aggressive multiple hypothesis correction. It makes it harder to find weak signals. So what's the benefit of doing this?
It includes splicing patterns. So if you had excluded parts of the sequence, that wouldn't be included in the group.
Yeah, so it will throw out, or well, in this case, it wouldn't necessarily remove the splicing. It would actually try to translate the introns.
Oh, you don't transcribe it first?
Not by default with how this works. You'd have to do some extra work if you wanted to do that. It just tries assuming the entire thing is an amino acid sequence.
You don't have to worry about different codon biases if you're comparing between humans and mice.
Yeah, so in a lot of cases, the same codon will code, or different codons will code for the same amino acid. And it's really an evolutionary issue that these codons can evolve, or the nucleotides can evolve more quickly than the amino acid. Because you have some redundancy in the codon alphabet, so you have multiple codons for individual amino acids, your nucleotide sequences evolve more quickly than the amino acid sequences in general. So if you're looking for the translation, you have a good chance of detecting more distant homology. The amino acid sequences will show similarity over longer time scales than the corresponding nucleotide sequences. So you might be able to catch some more distant homology than you catch just running BLASTN. So any questions about that? There are some more specialized blasts. I'm not going to dwell on these, but for example, there's PHI-BLAST, which allows you to throw in a kind of extra pattern to say if you're looking for blast hits only that match what's called a regular expression, so a kind of pattern in a sequence that you can specify. And this can be useful if you know that your match has to contain some particular residues. So if you know it has to have a glutamine followed by between 1 and 100 residues, followed by histidine, another loop of 1 and 100 residues, followed by an acidic residue. If you know something like that has to be in your match, maybe because it's an enzyme and those things have to be in the active site, then you can cut down on your search quite a bit. That will help you by speeding up your search, and it will help you find more distant homologs because it's reducing the problem of multiple hypothesis correction. The fewer things that might match, the less you have to correct for that, and so the less significant the p-value you need to get a good e-value. There's also PSI-BLAST, one that is kind of a blast that tunes for alignment to a particular family of proteins. Again, it can be a good way of catching more distant homologs. But as I say, these are things you guys can play around with when you work with the actual tools. We're not going to dwell on them. They're just kind of special versions of the main blast. The one last thing I wanted to tell you about is a bit about multiple alignment algorithms. So multiple alignment is computationally a very different problem than pairwise alignment, that is alignment of just 2 sequences. So pairwise alignment, I told you, has algorithms where for reasonably large sequences we can be guaranteed that we get the best possible alignment. You won't necessarily use those algorithms, but they exist. And when you have a problem like this, we would say that that is a tractable problem. It's possible to get the best possible answer in a reasonable time for very large sequences. Multiple alignment is what's called an intractable problem. And what this means is that if you have more than a few sequences, there is essentially no practical way of finding the best possible alignment. More formally, what it means is that you pretty much have to try every possible way of aligning things, or at least you have to try an exponentially large number of possible ways of aligning things. And when you have a problem that is intractable, what that's generally going to mean is that you get a lot more heuristic algorithms and a lot more work trying to figure out what the best practical way to do this is. So when you have a tractable problem, very often people will just solve the problem, you put out one tool, and then it's solved for good. It's not quite the case with sequence alignment because the sequences have gotten so big. When you have an intractable problem, there's always going to be room for improvement, so you tend to have a much harder time finding the one best tool that's out there, and multiple alignment is an example of this. There are a lot of multiple alignment tools. If you're trying to solve a hard multiple alignment problem, it's not obvious what's the best tool, so that's something to be aware of. There are always going to be improvements in multiple alignment algorithms. If you're having trouble with your multiple alignment, you may just want to do a search for other tools or algorithms that are out there, and you may find a better one for your problem than the one you're working on. But there are a few things that people tend to do in this case, and there's a very popular class of heuristics for this, which is another version of that kind of divide and conquer idea known as a progressive alignment. So a progressive alignment is a way of trying to solve the hard multiple alignment problem by breaking it down into the easier pairwise alignment problem. And it does this in a heuristic way, where heuristic means that it's not going to get necessarily the best possible multiple alignment solution, but it will get something that we hope is a pretty good solution by using the fact that we can optimally align pairs of sequences. And the way it works is basically as follows. You take your set of multiple sequences, and you try aligning all of the pairs of sequences using our much better pairwise alignment algorithms. So you align blue against red, you align red against green, blue against green, and you see which gives the best alignment. And maybe what you'd end up deciding is that blue against green is the best alignment. Those are the two most similar sequences. So what you'll then do is replace this pair of sequences with what's called their consensus. There are different ways of defining a consensus, but basically what it means is kind of the average of those sequences. Or a representative sequence you can throw in place of these two. It may be literally a sequence, it may be some more complicated mathematical object that captures the uncertainty in some cases, but basically something that you throw in as kind of a virtual sequence now to represent the alignment of these two. And you then repeat the process. If we had 10 sequences, we would now be down to 9 sequences, so 8 regular sequences and a consensus. In this case, we're down to 2 sequences, so at this point all we need to do is align those two. There's only one possible best alignment. You throw those together, you replace that with the consensus, and then you kind of go backwards in this tree of alignments that you've built up, and you insert the gaps in the sequences that were built up in those pairwise alignments. So if you go down the tree, throwing in these gaps, you end up building your multiple alignment. Just figuring out everywhere a gap was thrown in a sequence in building these pairwise alignments, that will give you your multiple alignment. It also incidentally gives you this tree of how these sequences were put together to build your multiple alignment. And that gives you a very kind of crude version of a phylogenetic tree. So an evolutionary tree of these sequences that is more or less predicting how these might have evolved from one another and what some of the ancestral sequences might have looked like. I will say this is not a particularly good way to make a phylogenetic tree, and we'll see later in the term better ways to do this, but it's a way that's often used to kind of get a quick and dirty phylogenetic tree. Any questions about any of that? Okay, so that is the idea behind a class of standard multiple alignment algorithms, and there's one particularly popular version people often use called the clust-LW algorithm. We'll see an example with clust-LW server a bit later. There are improvements beyond that, so it's kind of an older algorithm. There are better ones out there, but it's a good representative that many people who do multiple alignment have used. So what I want to do at this point is go to the more hands-on portion of this and show you how some of these things work online by going to some actual tools for alignment. What I'm going to do is open up here the NCBI BLAST web page. And I'll just show you here. This web page is found here. You can get this off of lecture slides. So ncbi.nlm.nih.gov/BLAST. That will take you to a BLAST tool that's linked to all the NCBI sequence databases. So it's linked to a lot of the useful things we would want to use here. And if we scroll down on this page, what we can see is that all those versions of BLAST I told you about are here. So we've got nucleotide BLAST and protein BLAST. There are different versions of these. I'll say a bit more about those shortly. BLASTX, TBLASTN, TBLASTX, and so forth. There are also some more specialized versions of BLAST. I'm actually going to start with one of these, which is basically a way of turning BLAST into a pairwise aligner. BLAST is usually used to search against large databases, but for illustrative purposes, I'm going to pick this align to or more sequences thing, which just sets it to do the default of looking at 2 sequences you supply to it. And what we would get here then is a page where we have room to enter these sequences. And if you go to the Blackboard page, you can see I put up some sequences if you want to try to duplicate what I'm doing here. And what I'm going to do is try to cut and paste these sequences into those boxes here. So I'm going to copy that one, paste that here. It's the query 1 sequence, and I will copy in the query 2 sequence. And paste that one in here. You can see that we have a few different options for what we might want to do with these sequences. So there are 3 different algorithms we can choose from here. The basic BLASTN I told you about is this one here. That will actually tend to be relatively slow as these algorithms go. And there are other ones that are tuned for running more quickly, but also having less ability to detect distant homologs. I'm going to set this on BLASTN for the moment because we're just looking at 2 relatively short sequences. Generally, you want to stick with the ones tuned for highly similar sequences unless you know you need to find distant homologs, because when you're looking against a whole database, it's going to run much more slowly if you pick the BLASTN versus the MegaBLAST. Well, I'll just try blasting here, and not sure what happened there. Hopefully this will fix itself, or we can go back to the slides to do this. So this is always the risk of trying to do this kind of thing in live lecture. These tools don't always work, but we've got it going now. And what this is showing is basic visual code showing us that we have a nice alignment between these sequences. This is basically saying that we've got a good p-value alignment in red here. I'm showing the region of the sequences that align. We've got something I'll just briefly explain called a dot matrix view or dot plot. This is often a good visual way of seeing for fairly long sequences where you have good alignments in them. And what this is showing is for any particular position in the sequence, it puts a dot there if you've got a little bit of nice alignment between your 2 sequences. So this is one sequence running along this axis, another running along this axis. When you see a long line like this running through the sequences, what that's telling us is there is a long piece of nice alignment in those sequences. We can see that there's this little break in that line here. Can anyone tell me what that might represent? Yeah, so that would be a gap. What this is showing is that basically there's a piece of sequence here in this one that doesn't align to anything in this one. So you get the sequences kind of slide in like this. Sometimes you'll see a line that runs in the opposite direction, so diagonally from upper left to lower right. Can anyone think of what that might mean? That would usually mean that there's a reverse complement match, so that one piece of sequence is aligning to the reverse complement of something in the other sequence. But that's basically the visual code that's used here. It tends to be good for longer alignments. More often what we would want to look at is the detailed information we see here showing exactly what aligned between the sequences. So there's kind of a visual code where you're seeing the two sequences that align, the ranges of those sequences, so often you're not going to be seeing the entire things aligning. BLAST is a local alignment tool. And then anywhere you have a match, it will put this little bar here, so you can kind of quickly see there are a lot of bars, you've got a good match. If you've got spaces here, that means a mismatching base. We also get some nice quantitative information we can use to make sense of these things. So you should be able to see here, we've got some basic scoring information on how good this match is. Let me blow that up a bit more. So the most important thing we usually care about here is this thing called the expect. And expect is just what I had referred to last time as an e-value. So expect is the multiple hypothesis corrected significance estimator. It's saying, on average, how many sequences would you expect to have an alignment at least as good as this one? This is saying 4e minus 142. That means 4 times 10 to the minus 142. So is that a significant alignment? Yeah, so that's a very small number. That means it's very unlikely this would have occurred by chance if we didn't have a real alignment. We also get measures of the percent agreement. So that gives us a more absolute measure of how similar these two sequences are. And we get a couple of things here under the score. The thing called bits here is a kind of disguised version of the p-value. That's not usually what we want to look at, but what this is saying is that essentially the p-value is 2 to the minus bit score. So the p-value for this alignment is 2 to the minus 489. It's an information theoretic way of encoding very small numbers like this, small probabilities, but that's telling us that this is also a very significant p-value. And the number in parentheses here is the alignment score in terms of matches and mismatches and all of that, so the actual match score. Usually we don't really care about this, but it's there if you want to know it. So that is basically the information you get from these alignments. Any questions about any of that? Okay, so generally, as I said, you're going to want to use these things for searching against a database. Let me go back to the start here. I will pick, in this case, the nucleotide BLAST. And what I'm going to do is now turn off that align 2 or more sequences thing. Make sure that's not on. I'm going to pick out the third query sequence here, and we'll do a search for that sequence. And I'll point out a few other things we need to pay attention to. So one of the things we would often care about in doing an alignment like this is the database we're searching against. So here, this is searching against nucleotide collection NRLP. That basically means search against everything in BLAST, except that it's not going to search twice against sequences that are exactly the same. That is a very big database. Often we want to be a bit more focused. So we might pick, let's say, RepSeq RNA. RepSeq RNA is a curated database, so it's going to have a nice human-checked transcript for a large set of RNA genes. So that might be something more focused we'd want to look at if we were trying to figure out what genes were present in the query we were looking at. So we can keep that there. I will leave this on MegaBLAST, so we want to search for relatively similar sequences. I'm going to open up this algorithm parameters here. So the algorithm parameters is giving us some additional information that relates to how the BLAST algorithm works. So some of this, the expect threshold, is just saying how high of a p-value will you tolerate. Usually 10 is a bit more than you want. You could maybe make that 0.01 or so if you only want significant things. The word size is the k-mer size. So what might be the advantage of making the k-mer size larger than this? It's set to 28, but we could make it larger or smaller. What might we get out of making it 128 instead of 28? Yeah, so if we make the word size bigger, it's only going to find closer matches generally, and that's generally going to make it run a lot faster. So as a general rule, the bigger the word size is, the faster it will run. What do you lose by making the word size bigger? Why not just make it huge in every case? You'll miss the more distant matches. So if your word size is big, there's more of a chance you're going to miss a real match that you didn't want to miss. So we can just leave this. That's the default filter and masking set of things here. So this is set to filter for low complexity regions. And low complexity means repetitive pieces of sequence. So AG, AG, AG repeated 100 times is considered a low complexity sequence. And this is often something you want to have checked if you're looking for nucleotide sequences. So to see why that might be the case, imagine I had a sequence that had a string of just A's, like 100 A's in a row. I'm going to assert that it would be a really bad thing to turn off the low complexity search and search your RNA database against that query with that long string of A's. Why is that? Yeah. Yeah, so if you have RNA sequences, eukaryotic RNAs always get a poly A tail attached to them. Often those tails will end up in the databases. Sometimes they'll be cut off, sometimes they won't. So if you don't check that thing to hide the low complexity regions, it will find those poly A tails, and then you're going to get lots of spurious matches. It'll take forever, and you'll get really useless data out of it. You can also hide sequence-specific repeats. So the human genome, or most eukaryotic genomes, are mostly repetitive regions. Usually if you're searching with a big piece of human DNA, you want to hide the human repeats, or all you'll get are millions of matches to those repeats. So, but I'll just leave that as it is. Just point out this one more thing while we let it search. So this is the information on the scoring function, and we can pick different options there for match, mismatch scores, for gap of penalties, for some of the more complicated ones. So again, that's something we can play with if we know we have reason to think that a particular kind of match is what's important to us. So if we know that we're looking for very similar homologs, maybe we want to make the mismatch penalty high, for example. Right, so this has turned up some results. We can see there are a range of qualities. We've got a few high-quality results, some lower-quality ones, and we can scroll down and see what some of these look like. So we see a quick summary of a few nice alignments here. Which of these are significant? So how many of these would you say are significant?
Is that the first three?
Yeah, so the first three have low e-values, less than about 0.01 might be a reasonable threshold. So these are probably the significant ones. These ones you'd say are very likely just false positives. And otherwise, it's pretty much the same as the output we got when we looked at the pairwise alignment we saw before. So basically, you get the same kind of information about the quality of the alignment and a general view of what we see in that alignment. I will just point out briefly, there are some other nice tools associated with this. You'll get a chance to look at these in a bit more depth in recitation, use them yourselves. Let me just show you kind of briefly an example of what you can do. So if we wanted to know how these homologous sequences might be related, we can check a few of these, and we can ask for a distance tree of results and get a model of a potential phylogenetic tree built from these alignments. So I'm just going to switch this to a slanted tree so you can see it in a bit more detail. In this case, at this level of zoom, it's hard to see. Try zooming out a bit more, and you can kind of see that we get a tree here. Sometimes you need to expand some subtrees that's kind of hidden from you, and we can see where our query sequence sits relative to the things that seem to be nice alignments to it in the database. So any questions about any of that? Okay, so I also want to briefly show you protein alignment, because in some ways that is a bit different from nucleotide alignment. So I'll just go back to BLAST again, and I will pick protein BLAST. Here again, you can paste in a sequence. So let me pick now the query 4. It's a protein sequence. Paste that in here. In this case, I'll leave most of the options the same. I will just leave the default NR, so again, that's the big database, everything in GenTech. I'll leave the algorithm on BLAST-P. If you just want to briefly point out, the algorithm parameters are a little different in some important ways. So we have a much shorter word size. Can anyone think of why that would be the case? Basically, with amino acids, we've got a much larger alphabet, so each character in some sense is much more informative. You don't need as long of a word to figure out if you've got a good match. We've got this Blossom 62 checked here. What is that? That's our substitution matrix. So we have options available there. If we wanted to find very closely related sequences, we might switch that to PAM 30 or Blossom 90. If we wanted to find very distant sequences, we might switch it to PAM 250 or Blossom 45. And I'll just leave it on Blossom 62. And then we have various other options available. Usually you don't need to worry about the low complexity for protein searches. There are exceptions to that, but usually you don't have nearly as much repetitive sequence in a protein as you do in a nucleotide sequence. So we can run that. We can come up with a result here. In this case, it's showing us that there's kind of a quick search you can do to figure out there are some conserved pieces of sequence here, some what we would call protein domains. We'll return to that topic next week, but it's continuing the full BLAST search, and hopefully will show us a result shortly. Sometimes these things can take a while to run. Hoping this one doesn't take too long, but yeah, so here it's giving us our results, and we can see something pretty similar to what we saw before. Various qualities of alignment. You can look at our e-values, see there are quite a few significant alignments here, and I just want to illustrate one difference between these protein alignments versus nucleotide alignments. It's a slightly different visual code to show you if you're looking at a good alignment. Where the two residues agree between your sequences, you'll see a repeat of the letter. So we see an F here, phenylalanine aligned to phenylalanine, so you get an F between them. But the disagreements are sometimes spaces, sometimes pluses. And that's designed to give you a little more nuance in seeing if you've got a good match. If it's a space, then what that means is that it's an unconserved alignment. You have two chemically dissimilar amino acids. A plus means they're not the same, but they're chemically similar. So it gives you a little better ability to see when it does. By looking at this, this is a pretty good alignment, even though it has a fair number of mismatches. So, any questions about any of that? All right, so I just wanted to show you one more thing, and that is ClustelW. Just show you briefly what a ClustelW server would do. There are lots of different places that have ClustelW servers, and we can take one example of that. So this is the EBI ClustelW server. EBI is the European Bioinformatics Institute. It's sort of an analog of NCBI in Europe. And we can take their server. What I'm going to do here is pick this query 5, and the query 5 contains multiple sequences. So pick this set of multiple sequences. I will just point out here that the format of this is kind of important. Often when you're working with these algorithms, you need to be very specific about how things are formatted. So you might see this structure here where we've got a greater than sign followed by a symbol with no spaces in between. That's actually important. The algorithms will look for that, and if you don't have that exactly in that format, it may not be able to figure out what you're showing here. But basically, you have the greater than line, a name for this sequence, then on the next line and following lines, the sequence itself, and it will keep reading sequence until the next greater than. So we can have a set of data of entries like this, and that's known as a multi-FASTA format. It's a common way of representing these multiple sequences. And what we can do is use this ClustelW server, and it uses both a multiple and a pairwise alignment. So it's using the pairwise alignment for that progressive alignment strategy. You can pick some options for that. Maybe say we want a Blossom matrix. It doesn't give you as many choices as we get with the BLAST server. You can pick Blossom also for the basically optimizing the multiple sequence alignment. And what I'm going to do is submit this, and this should hopefully quickly come up with a multiple alignment for our sequences. Again, sometimes takes some time, but here we've got a multiple alignment. Let me blow that up a little. And what we can see is this has done a nice job of lining up all of our sequences to show where there is conserved sequence between them. In some cases, we'll have exactly the same residue across all of our sequences in one position, and it will visually code that by putting an asterisk below them. In some cases, we will have not exactly the same thing in each sequence, and then depending on how well conserved it is, we'll either have a colon, highly conserved, a period for low conservation, or a space for no conservation. So again, you can kind of quickly look at what's being shown here and figure out if you're looking at a pretty good alignment. This also gives you ways of looking at your phylogenetic tree associated with this. So this is just a text version of a phylogenetic tree and a visual version. And if we put this on real here, gives you some measure of the branch lengths, basically an idea of how these might be related to one another and how they might have evolved from common ancestors. So again, in recitation, you get to play a bit more with some of these concepts, to look at how we would do these alignments and try out some of these tools. So we're out of time here. Are there any last questions before we break for the day? Okay, so if I don't have your homework, please make sure we have that now. And you TAs can answer any more questions.